You're listening to Idea Collider, a show that explores the world of asymmetric learning. On this show, I will sit down with pharmaceutical experts and business leaders to discuss how to embrace uncertainty and the different learning style that follows. I'm your host, Mike Rea. Let's get into the show. So welcome to my last recording for Idea Collider. I'm delighted to revisit a recording that I made with Reagan at JP Morgan, where sadly the audio kind of let us down, but it was such a fascinating interview. I didn't want to let it drop. So I'm delighted that Reagan was kind enough to agree to do this again. Reagan, welcome back. And it was great to have you on Idea Collider. Could you, for the first time, tell folks about who you are and the kind of founding of Anocca?
Yeah, so I'm Reagan Jarvis. I'm the science founder, CEO at Anocca. We're a Sweden-based biotech that's focused broadly on T-cell biology. At the heart of the company, we've built a technology platform which is analytical in nature. So it's a cell biology platform that allows us to map genuine T-cell targets, so these HLA-presented peptides, generate very quickly and cost-effectively big sets of T-cell receptors that recognize those targets and then do very sophisticated differential analysis on the quality and the safety of those TCRs and even directed evolution of TCR potency. And we've directed that primarily into TCR-T cell therapies for oncology. We've got our own manufacturing facility here in Sweden and deployed a novel non-viral gene editing process for autologous TCR-T cell therapies. First clinical program up and running, which is here in Europe, 8 centers for phase 1 across 4 countries. That's the first non-viral gene-edited T-cell therapies to reach the clinic here in Europe. That's mutant KRAS targeting, exciting target, and is focused on a single indication, pancreatic ductal adenocarcinoma. So we're pretty excited about that.
Tell me, just remind me of your kind of scientific origin. What was your lab time based on?
Yeah, so I'm a Kiwi, New Zealander. Originally, I trained there in biochemistry, cell biology. So originally enzymology, and then moved into cell biology, signal transduction research. And that's super fun stuff. That was very niche, like electron exchange reactions in signal transduction. Pretty small field, but very fun method development type of research. And then a couple of postdocs, one in clinical immunology, looking at T-cell trafficking in inflammatory bowel diseases, and then back to my first love of signal transduction for a couple of years in Germany at the German Cancer Research Center. And then came up here to Sweden 2014 to start up Anocca, which was literally an idea on paper, backed by my co-founder, Mikael Blomqvist. And we've built the company out from an idea on paper to what it is today. It's taken us 12 years to get here, but it's been a fun journey.
Yeah. Well, tell me more about that, because, to a lot of people, starting a biotech, being CEO of a biotech, would be kind of mind-blowing, but what was your inspiration?
Well, it's less inspiration as frustration. So the idea for the company came from working with T-cell biology and coming out of a more biochemistry, cell biology, molecular biology background, getting to T-cell biology back in the, what was it, late 2000s. It was frustrating that you couldn't really do anything about precisely mapping targets and generating TCRs and looking at how they actually act in a cell biology context. You're kind of trapped with doing binder screens, phage display, and which doesn't really get to the crux of the problem, which is how do these cells process and present, target cells process and present the HLA-peptide targets, and how do T cells recognize those targets. So it's not like an antibody ligand interaction, although the molecular systems are similar. It's actually the cell communication. So that frustration of not being able to do very specific things with the biology, came the sort of idea, hey, we can just systemize cell-based assays to tease that biology apart and look at that fundamental cell biology, which is so key to T-cell biology generally.
Yeah. You raised a bunch of money at the end of last year. What do you think about the world's view of confidence in, your vision for TCR-T in solid tumors especially?
Yeah, I mean, there's been a sort of low ebb in enthusiasm for T-cell therapies in oncology. Back 2017, we saw the first CAR-T approval, and then a number since then. And CAR-Ts have been transformative in hematological malignancies. Absolutely stunning success, really. I mean, some time to get the commercial up and running and the sort of manufacturing scale, but shown it can be done in an autologous cell therapy setting in oncology. Now, I think the low enthusiasms come from the expectation that success in hematological malignancy for CAR-T would translate to solid tumors, and
Yeah.
it really hasn't. Our view on that, all the way back in 2014 when we started the company was, hey, there's just not that many, if any, really valuable solid tumor targets for CAR-Ts. The success of CAR-Ts in B-cell malignancies have been targeting the entire B-cell compartment with CD19 BCMA so far.
Yeah.
And that's tolerable. I mean, you can manage B-cell aplasia, you get rid of the malignancy, you get rid of all B cells as well, and that's tolerable in a clinical setting. So, but you can't do the same for solid tumors. The analogy I often use is if you treat liver cancer, you can't get rid of the whole liver. So you can't target a liver lineage marker. So what you need is something that's much more tumor specific. And that's, there are so many targets available there. The caveat being that there are these very complex HLA-peptide targets that are highly segmented across patient populations because of the genetics of the HLA presenting complex, which is, gene family is the most polymorphic in humans. So there's a lot of diversity of how those peptides are presented, and of course the diversity of mutated or dysregulated sequences in those tumors. So there's that fundamental sort of complexity of the target space, and then we would assert as well that the complexity of the targeting receptors themselves, which are much more nuanced and subtle than an antibody or a CAR engagement of a target, and these sort of complex, somewhat promiscuous, but extremely sensitive receptors that you need to work through that biology. And that's where the target space is. And, we've seen some success in TCR-T. We saw an approval from Adaptimmune. It didn't work out commercially because of the niche market opportunity. But we're seeing Immatics move forward with a very decent product in melanoma and some good data in ovarian and other indications in that sort of basket setting. So, we're seeing the opportunity that there's a target space to leverage. Our platform is about leveraging that target space very time and cost effectively, not just in discovery, but with our manufacturing approach to mobilize multiple first-in-human products trials very rapidly.
Yeah, well, you've got I'm going to say, an interesting sort of fully integrated platform, right? So it's more than just the molecules and drugs that you're putting out there, novel biotechnologies, custom software, in-house manufacturing. How's that kind of vertical integration giving you kind of, I'm going to say, competitive advantage with all of what you described?
Yeah, it comes back to time and cost to mobilize new products and product candidates. Like I say, the target space is tumor selective, and it's very clear what you have to target at the end of the day. It's about how you move through that discovery and how you do your product development preclinically and mobilize those to actual first-in-human trials. So that vertical integration per se is about the systematic mapping of targets, generation of the targeting receptors, characterization optimization, but also marrying that with a scalable product manufacture. So we've established our own facilities here, deployed this non-viral gene editing process, which is partly about making reliably potent products because we're doing TCR replacement. We get rid of the endogenous TCR, deliver our therapeutic TCR, but it's also just much more time and cost effective to build novel product candidates because we're not having to develop a new viral vector every time we've got a novel TCR. It's a gene editing process with the editing reagents. The gene editing reagents are universal across all products, and we need a new DNA template encoding our novel TCR. So it's not 18 months plus to develop a new viral vector and a couple of $1 million. It's months and hundreds of thousands of dollars just to get that novel input to manufacture. So it's this is a modality where you need to drive scale. You need to access the target space, and to do that, we've decided we need not just the cool technology on discovery, we need the robust technology for manufacturing those novel products as well.
Yeah, so end to end. So in 2025, you got a sort of CTA approval and started recruitment for, is it VIDAR or VIDAR-1?
Oh, it's a Swedish word. I think it's VIDAR.
VIDAR, okay, yeah, that makes sense. That was the first non-viral gene-edited TCR-T in Europe. So that kind of desire to turn into a kind of real thing, can you describe the kind of hurdles and the platform and the challenges that were there to get to first in human?
Well, I mean, technically, people know how to gene edit cells. So there weren't too many technical barriers there. It was was operational, right? I mean, we actually set up our GMP facility around a lentiviral-based T-cell manufacturing process, and we converted over to the non-viral gene edit for the reasons that I described. But it was just good applied science. The regulators were great here in Europe, good dialogue, and honestly, very few questions, RFIs on the clinical trial application relating to, let's say, the quality of the product and the preclinical package. It was all, nitty-gritties, RFIs around, the manufacturing inputs. So that was a fairly, let's say, relatively straightforward process in the grand scheme of things. And got a lot of experience in the company around not just, GMP, facility design and so on, but also just the fundamental gene editing technologies and handling T cells. So there's some challenges, of course, but it's relatively straightforward and painless in the grand scheme of things.
Yeah. Okay. Talk me through kind of one, because you've got this kind of, talk me about focusing on KRAS mutant pancreatic cancer. And, I'm a big fan of what I call asymmetric learning. So your kind of umbrella design, testing multiple candidates, how does that allow you to learn more efficiently?
Yeah. So the history of TCR-T is interesting, right? It's been around for a long time. There's been some success, in academic settings and in commercial settings as well. But the trial designs there have largely been these large basket trials where you recruit all comers essentially across a number of indications with the target HLA and gene expression of interest or target expression of interest. And that's challenging, operationally. You're having to manage all these different indications, different prior lines of different standard of care before they get the TCR-T. And it just creates noisy data when you get to phase 2A. Difficult to interpret, not just time and cost inefficient, but let's say data inefficient, and be able to interpret that phase 2A or phase 2 data. Our approach is let's focus down onto single indications and bring more TCR-T assets to bear. And because KRAS is a bit more complex because there's multiple mutation forms, but each of those are also presented in different HLA restrictions. So it's a personalized therapy by definition. You have to match the product to the individual patient with target expression. And the value of bringing multiple assets is that, frankly, you get more bang for your buck on your screening program.
Okay.
You're activating these clinical centers, you're screening these patients for your HLA and mutation status, and you can personalize those to a library and an umbrella design. It also gets you on that single indication focus, cleaner data, but also the opportunity to design your program to integrate more closely with existing standard of care.
Yeah.
Pancreatic ductal adenocarcinoma trial, VIDAR-1, is quite a neat design where we're actually recruiting patients at diagnosis, collecting the leukopak, so the patient T cells that we do the autologous manufacture from, and then those patients actually come off protocol and go into standard of care. So in Europe here, that's typically FOLFIRINOX or some other chemotherapy regime, which takes, give or take 16 weeks. So we've got this luxurious window to do our manufacturing in the cryochain, get those cells in the cryochain. And we actually treat patients that are in partial response or stable disease from the standard of care. And we're treating immediately as a consolidation in that patient population. We make product available for non-responders under compassionate use. So we also, ethically make that available, but also get to collect data informally in that direction as well. So we just think it's a great way to get patients intervene early, collect that relatively fresh T cell compartment to manufacture from, and maximize chances of success. The patients get these chemo regimens because they do bring value. That chemo is just so toxic, you can't keep giving the patients additional cycles, and we want to consolidate the benefit the patients are seeing. So, interesting. So it's a design, intervening early, lots of benefits there.
Yeah. Well, I'm interested in maybe the kind of start on the endpoint. So, collection of biomarkers, are you looking at anything interesting there? And on the flip side, the endpoints, because you've got this kind of old set of endpoints in this disease, are you looking at anything novel there as well?
I wouldn't say it's terribly novel. I mean, it's all the standard stuff, right? Resistant, we're looking for T cell engraftment and CRS and so on. But I guess the more modern biomarkers we're looking at there, DNA, so circulating tumor DNA shedded from the tumor. It's kind of a biomarker for on tumor activity. You'd expect that to spike early days as the tumors are getting beaten up by the TCR-T product, and then ideally you'd see that diminish over time as they're declaring tumors. And you've seen that in a number of immunotherapy settings, including a personalized vaccine setting, where that's been an interesting biomarker.
Interesting. And you've got a bunch of targets as well, discovery through early clinical targets like P53, PRAME, MAGE, more beyond KRAS. This sort of systematic TCR mapping platform. Can you build a sort of deeper, better library than anyone else can with that focus?
We'd like to think so. The premise of the whole discovery platform is we map HLA-peptide space from tumor-restricted gene sequences. So you take a PRAME, a MAGE, a P53, a KRAS, and you basically determine which HLAs present peptide from those targets.
Yep.
And you make big libraries of TCRs, in our case, only from healthy donors, and you use those TCRs as a source of truth. You validate the target, you get a sort of wealth of TCR data to do differential analysis on those individual targets, do some orthogonal assays across tumor cell lines, and sometimes mouse models, ex vivo models, to get a feel good about how your target and your TCR combo looks. I'd say the bigger picture there is we make TCRs to validate targets, so we have a lot of preclinical assets. Just the systemization about how we characterize those and in some cases optimize those TCR frameworks through a directed evolution process is that it's very rapid. We can go from gene sequence of interest to a TCR-T product that we can put into the manufacturing framework in under 12 months. It's very parallelizable and it's very cost effective. We're talking hundreds of thousands of dollars, not millions of dollars to make a new asset. So that speaks to the scalability, which is achieved by how we've systemized these cell biology assays, but also the fact that it's extremely precise and biologically relevant data. We're actually looking at the T cell biology, not proxies, not readouts from primary cell assays or binding assays, affinity maturation assays. So also a huge amount of data. You mentioned it as well. We've got a software ecosystem we've built ourselves in-house, which is kind of an enterprise software to run the R&D organization. But at the heart of that is the big database collecting all this beautiful cell biology data with a huge amount of diversity around this HLA-peptide TCR problem that we look at so closely. And that we're building machine learning models from that data, which can accelerate the discovery process for these personalized TCR-T development. We think there's a fairly clear line, although not trivial, to make this fully individualized. So doing in silico target mapping, TCR candidate generation that you can then do on a patient by patient basis, which we think is part of the future, but is what you require to get there is relevant data at high depth, around this central problem, HLA-peptide TCR biology.
Yeah, I'd be interested in maybe sidestep into, where you think the FDA sits in these kind of N of 1 studies that you might be headed towards.
Hey, there's plenty of precedents there, right? I mean, personalized or what we call individualized cancer vaccines been around a long time. They're basically N of 1 products. So, and you've seen frameworks put up around manufacturing platform designation from the FDA, that you can get sort of master protocols for your manufacturing platform, not just in vaccines, but in gene therapies and more recently in cell therapies. So we think that the regulatory paths there are emerging, and I don't think they're going to be the major bottleneck here. I think the bottleneck is understanding and systemizing the discovery biology and integrating that with the scalable manufacture. At the end of the day, if you demonstrate efficacy for these products and you can make them at a reasonable cost, then the regulators are going to be along for the journey. And then they're really playing ball there. They're trying to put up these frameworks and be engaging and map pathways for these types of very advanced products.
Yeah, and then maybe on the other side of that, the kind of commercial landscape, you mentioned the sort of challenges in the past. How do you see that evolving? Do you see folks like Kite and Kyverna, a bunch of others sort of, building a better landscape?
I mean, look, the autologous cell therapies work. The CAR-Ts, they work unbelievably well in oncology. It's been a journey to reach reasonable scale there. I think Carvykti from J&J hit $2 billion in sales last year, which is a good product. Kyverna and others looking at autologous CAR-T in the autoimmune setting, can be done. A lot of energy around allo, donor-derived and iPSC-derived allo. Obviously, a lot of recent deal-making around in vivo CAR-Ts. We think there's a path for all of these therapies. We think in the very near term, autologous have been demonstrated repeatedly to be both efficacious and safe. That's where we are as a company. We do have collaborations for iPSC-derived allo products. We develop constructs in-house for in vivo TCR-T. But for us, the core of the company is analytical in nature. We map targets, we make targeting constructs. That's the fundamental barrier to entry. In the longer term, we're pretty agnostic to where the manufacturing tech goes. At the end of the day, I think we updated this analysis recently. We count, I think, over $200 billion invested in T-cell therapy manufacturing technologies and infrastructure over the past decade. That buys you an awful lot of innovation and know-how. Where the field is that we lack targets and novel targeting constructs to mobilize novel products. So you can't drive scale for any manufactured product unless you've got novel products. And there's only so much room for more CD19 BCMA CAR-Ts in oncology, probably no more room. And there's going to be limited room there for autoimmunity as well. So we need novel targets, we need novel targeting constructs, and that comes from HLA-peptides and TCRs. That's where the target space is. You just need to mobilize this fundamental sort of discovery biology to achieve that.
Well, it's a $200 billion island waiting for some kind of evolutionary mutants to kind of come along.
It's also, you can look at the vaccine space in a very similar light. You count hundreds of vaccine formulation platforms, whether they're viral or LNP, mRNA LNPs. I think the same challenge exists. What are we going to encode in those vaccine platforms? Our opinion very strongly is they're T-cell targets. Humoral immunity, we understand. We can encode antigens that will provoke antibody responses. But there's this fundamental barrier to solubilize, what are the T-cell targets to encode in these types of vaccine platforms? Broader applicability of our platform across infectious disease, autoimmunity, even allergy, because it's that fundamental question. What are the targets and what are your targeting constructs to put in all this beautiful cell and gene therapy manufacturing capability that we've got?
So, the autoimmunity, infectious disease kind of plays, I know you've explored it. What do you see as the kind of evolution from where you are, the kind of near-term focus on solid tumors into that space? What are the kind of steps between?
Well, we can't, as our chairman would say, boil the ocean. So we're focused on the oncology TCR-T programs. We've done preclinical validation of our platform in infectious disease and autoimmunity. And there's, like I say, a wealth of manufacturing technologies out there in both settings, whether it's TCR-T reg, whether they're prophylactic or therapeutic vaccines in infectious disease, or tolerizing vaccines in autoimmune syndromes, or even allergies. So that's for partnerships, the co-development relationships. I think the field is moving that direction. Like I say, a low ebb for cell therapies in oncology, and I would say a tentativeness to drill down into these extremely personalized cell therapies, therapeutics or prophylactics. I think the way through that is just to understand the target space better and optimize targeting constructs, no matter the disease area or frankly the modality. T-cell biology has the target space. You just need to break it down and systematically understand those targets and deliver the targeting constructs.
I thought you mentioned that you're sitting in Södertälje, Sweden, in the old AZ plant. Building a cell therapy company in Sweden with kind of global ambitions, you've got your kind of unique challenges, regulation, talent, manufacturing scale. What do you see as the kind of biggest lessons from the last couple of years as you kind of grow towards this kind of preclinical to dosing patients phase?
Biggest lessons? It's a good question, Mike. I think that's a hard one. The biggest lesson is probably clear communication of what we're trying to achieve. It's a complex space. As you hear, there's a lot of biology going on here, a lot of moving pieces and different manufacturing modalities. I think the lesson is, let's just be very clear about what our near-term milestones are. Set up those milestones and show people we're knocking them down. I think as any biotech company, that's what you have to do. But in such a, I would say, misunderstood space of TCR-T T-cell centric therapies in general. That's what's important to us, and that's the lesson. Just people have said to me, and I've said this on a podcast I recorded a few weeks back with someone else, is that I've been told time and time again, when you're pitching to people, whether it's a investor, potential investor, or a partner, organization, if you're explaining, you're losing. I've heard that again and again, and I really disagree. I mean, I think we need to sort of embrace the complexity of this biological space, explain what the targets are, what the targeting constructs are doing, how the product is manufactured, and what that does in the body, to show how and why what we're doing is different. And I think that applies to a lot of our peers, no matter the technology area. I think there's such a lot of, for lack of a better word, complexity in modern therapeutic development, right? It's the science stack and the technology stack is just so deep that you need to explain your positioning, or the biology and the therapeutic area you're in, before you can explain your positioning and your differentiation.
It's kind of interesting challenge in biotech, right? Because I mean, that explaining and losing kind of metaphor I've heard a few times, but it doesn't exist in AI or tech world, or some of the other places. That's literally the way that things go, is you need a different background to explain why you're better than somebody else.
Yeah, I think so. And I think in the AI space, by AI these days, I mean LLMs. I mean, people think they understand that, right? Because you can get your hands on ChatGPT or whatever model and ask it questions, and people feel like they intrinsically know what it is. I think that's different for most fields of biology. People don't really have a feel for what it is and what it does. So I think there's a fundamental difference there. People interact with tech all day, every day, right? Whether it's hardware or software. But biology, even though we're made of biology, people don't really have an appreciation for what it is and the fundamentals. So I think it's a good compare and contrast between tech and biotech. But these two fields are coming closer and closer together, and I think the synthesis of these fields is super important to how we're moving forward as an industry.
Yeah. What do you think would be the kind of one misconception about T-cell therapies or solid tumor immunotherapies that you wish more people, investors, clinicians, or the general public understood?
Yeah, I think it's just the role of T-cell biology, and specifically the role, how these, for example, in the solid tumor space, how there is a plethora of tumor selective targets available, and that biology is inherently different from, let's say, the biology behind antibodies. That antibodies, recombination, VTJ recombination makes randomized proteins, immunoglobulins that can bind to things they've never seen before. Great, that's a binding interaction. We've understood that for 30 odd years, and beautiful platforms arose late 90s, early 2000s to deal with that. Now, the difference with T-cell biology is that there are two cellular systems interacting with each other, molecularly, with HLA-peptide and TCR that looks similar to an antibody ligand binding interaction, but there's all these intricate cellular processes dictating each side of that equation, with hundreds of proteins, membrane dynamics, cellular processes at play. So it's really about understanding that communication between the cells and all the processes that are going on. And that's where our approach comes from. We're not trying to outsmart biology. We've built a cell biology platform. We plug in the analytes, HLA, target open reading frame, TCR, and we just let the biology tell us the answer. Does the TCR recognize this? Does this polypeptide get processed to appropriate peptides to be presented in this HLA, and so on? We just program the biology and let the biology tell us the answer. And I think that kind of approach is underappreciated, because in those dozen odd sentences I just said, there's so much behind it that is just like I say, inherently different to what people are used to thinking about in drug development.
Yeah, and you explained before this kind of idea of you guys being a very solid discovery engine versus a kind of traditional pharmaceutical play. That kind of fits with that vision, right? Of just understanding how things work better.
Yeah, and like I say, I mean, the space in general, targeted T-cell therapies, whether they're cell therapies or vaccination-like approaches, it's a recognition that it's complex, but by definition that they must be personalized or even individualized therapies, because that's how the biology works. So I keep saying it's about scale. You need to be able to scale that discovery, and of course, consequently scale your product development as well.
Okay, let's switch to maybe 26, because you've got recruitment ongoing. Sweden, Denmark, Germany, Netherlands. What should we expect to see in terms of milestones for Anocca this year?
Well, we're aiming to move all the way through phase 1 for our first product in, which is a KRAS G12V targeting TCR-T product candidate in the HLA-A11 background, which is a bit of a technical designation. It just says, hey, this is the HLA presenting this mutant peptide from KRAS. So we will ideally get through all phase 1 patients there, which is ideally only 6 patients in that pretty compact phase 1 dose escalation, and then see ourselves extend into a phase 2A under the adaptive trial design. We're also looking to get this year and then early next year at least 2 more CTAs submitted to start building out that umbrella program with additional products, additional KRAS mutations, additional HLA restrictions, which is, like I said earlier, it gets more value from our screening programs, gets more product into patients, but also demonstrates that we can achieve scalability and discovery and product manufacture, which that I keep saying is the key to developing targeted T-cell therapies. You need to hit that scale.
100%. And if everything goes as hoped, how do you see, maybe the next 5-10 years for Anocca? What do you hit market, within that time frame? And how do you see yourself in 2030?
Yeah, 5-10 years could, let's say 5 years, so I don't have to speculate too much. So in TCR-T cell therapies, basically every product is an orphan product. Because once you cut down to a disease and the genetic restrictions, you're an orphan product. So you can see pretty accelerated approvals here, even on the back of 2B data in principle as a provisional approval. So, in the next 5 years, we see definitely a potential to have marketed products from these first KRAS TCR-Ts. Would also say, hey, we're poised to do a number of deals around co-development alliances in other disease areas and modalities. Fundamental tech is built, software is looking great, machine learning coming along. That's how we accelerate targeted cell therapies across, cancer, but also infectious disease, autoimmunity in particular.
Fantastic. Okay, and maybe my final question, I'll let you get back to the day job. Go back to New Zealand. What one piece of advice would you give to kind of other scientific founders navigating the journey from sort of bench to clinic?
It's something I always say internally, and I seldom say it externally, but it is recognize the difference between academia and biotech.
Okay.
And what I say is, academics ask questions, biotechs build stuff. And
Okay.
you can't lose that distinction, because we've got a huge number of fabulous scientists here, and the temptation is always to dig into mechanistic understanding or do fun stuff, but you have to stay on task and you have to build systems, you have to build products at the end of the day that bring value. So it's about resisting the urge to do too much deep science and actually do the applied science and build things. I think that's the one piece of advice I repeatedly give internally here at Anocca, but I think it's a good perspective more generally across the industry.
Well, it sounds slightly ironic because you sort of are a biotech based on asking questions, right? So it's interesting framing.
Yeah, I mean, asking questions, but it's about asking the same question systematically. What is the HLA-peptide target? What's the TCR? What's the targeting construct you need? So basically we've drilled down to one question, and we're really good at asking it, repeatedly in a number of dimensions. I think the way to get there is, like I say, we've built the systems, built the assays, built the software, built the infrastructure, and built the talent internally to pull that off.
Reagan, it's always a pleasure talking to you, but what a great way to sign off on my Idea Collider time. Thank you for your time, and good luck with the rest of this year.
Thanks, Mike. Thanks for the good chat, and a privilege to be the last one.
Absolutely. Take care.
Thanks. Best of luck.
That's it for this week's episode of Idea Collider. To continue the conversation, visit our website at ideapharma.com. Follow us in your favorite podcast app so you never miss an episode. Don't forget to rate and review us on Apple Podcast. Until next time, I'm Mike Rea, wishing you great success.